Utmust, University of Toronto, Most Understandable Scientist Competition. My name is Richard Lopez, and I'll be hosting the event today. So we all know that, especially at a university, especially as graduate students, that research is very important. And being able to do good research, but also being able to present your research in a concise and understandable way is just as important, especially for people who are not doing this, people that you want to convince to fund your research, for example. And so today we hope that this contest can bring out that ability to summarize things in a very concise way amongst our students at UofT. And so today we're very fortunate to have graduate students from economics, from psychology, from the natural sciences like physics, life sciences, chemistry, social sciences, etc. And we're also very fortunate to have 4 judges with us today. First of all, I'd like to introduce Sylvan Nelson, who is Vice Provost in Academic Programs. Miranda Cheng, Director of Center for International Experience. Jane Freeman, Director of English Language and Writing Support. And Chen Shen, Vice President of Royal Ontario Museum. So I'd also like to mention that today's event has been co-hosted by the UTGSU, the UTCSSA, the Chinese Students and Scholars Association, and also the in particular the GSU's International Student Caucus, which is chaired by Yan Wang, and I'd like to ask Yan.
Good afternoon, everyone. On behalf of the organizing committee, it's my great honor to welcome all of the speakers and audience to join us on this initial UofT Most Understandable Scientist. And also special thanks to all 4 judges with a different background, but the same passion and care for our international graduate students at UofT. Actually, I was reading the news the other day about our newly UofT scientific contribution to the ranking among the world, which is surprisingly come to the 14th among the world. And I'm definitely very proud of it, for sure. But when I thought about my own research as a 30-year PhD student, I just wondered how much I contributed to this ranking. And can we contribute more? And there are over 2,300 international graduate students at the UofT. And definitely we are making amazing scientific contributions to our community, and most of which are at least needed to the general public. But we do have our limitations. For example, English as the second language, and sometimes as the third, preventing us fully delivering our ideas, exact ideas. So it's quite important for us to find a better way and polish our skills to present our exciting researches, and which is very critical to our success as a young scientist in our career. So it is my best hope and every organizer's best wishes to provide this platform to each international graduate student at the UofT, and not only for the speakers, but also for the audience. And I hope in this platform we can learn from each other the skills, and also the invaluable comments from all the judges and each other, the audience and the speakers, and to be a better, understandable scientist. And also, as a result, to contribute back to our UofT's fame in the future. So, I want to be an understandable scientist someday, and I am very looking forward to the upcoming 12 speakers, and also the invaluable comments and input from the judges and audience. Cheers, enjoy, and thank you.
When I moved to Australia, I saw for the very first time the Milky Way galaxy stretched across the sky. It was beautiful. The Milky Way galaxy is our own home galaxy consisting of billions specks of light because they are so far away, even though they're actually very, very large. For example, our own sun, our own star, the sun, is so much bigger than the whole entire planet. Our whole entire planet, which you can see in the picture there. Our sun is but a baby amongst the stars. The largest star that we know of, Canis Major, is 1,000 times larger than the sun. Look how small it is. Beyond the edge of our Milky Way galaxy, there is a lot of empty space. Our nearest neighboring large spiral galaxy, Andromeda, is 3 million light years away. This means even if we were to travel at the speed of light, it will take 3 million years to get there. Imagine all the empty space in between. Some galaxies are so far away that the billions of stars that they contain become so faint, it is as faint as a single star in our galaxy. So yes, as you look at the night sky, what you think to be a star might be a whole entire galaxy consisting of billions of stars. As a child, I looked into the sky, and it was constant and beautiful. But the constancy of the night sky in the universe is an illusion. In fact, the most violent processes in the whole entire universe occurs out there in space. The birth and death of these very, very large stars that we see. The birth of a star is the birth of light. It is the birth of oxygen and carbon and all the other elements we see, like the iron in our chairs. We are literally made of star stuff. So astronomers study galaxies in order to understand the conditions in which stars can be born. This is a typical picture of a galaxy. You can see there is an edge. Beyond the edge, there is nothingness. The reason why there is an edge to galaxies is because stars cannot form in what essentially is empty space. There is not enough density of material and therefore not enough gravity to pull stuff together into a dense ball and create a star. This is why it was such a huge deal and a huge surprise when in 2007 we discovered something crazy. We discovered that stars were forming in empty space, or what we thought was empty space. This is a picture of what we saw. To compare, I've overlaid a picture of what we think the size of the galaxy is. And you can see in all these regions, UV light. UV light indicates young stars, stars that have just been born. How can these stars be born in empty space? And if stars are forming now, were they forming in the past? If they did, why don't we see that? In order to find out, my supervisor and collaborators at Yale University built this. This telescope, the Dragonfly telephoto array, can see the faintest things in the universe. It can see fainter than any other telescope in the world. What we saw was crazy. We saw a whole bunch of old stars that extended way beyond the edge of what we thought was the edge of galaxies. Suddenly, galaxies got more exciting. They can be 3 times larger, and stars might form in the most unexpected places. For my thesis, I'm going to look at a lot of galaxies and see if it is true for all of them. And we might find that these hidden giants allow stars to be born in empty space. Thank you.
My name is Masuya. I'm from the Department of Physics. So I did my major in undergrad, biology and physics. But then I chose, when I went to grad school, to do physics, mostly because I wanted my life to be a little easier. However, I didn't want to give up on biology either. So when I asked my supervisor for a biological sample, he suggested muscle. And it was interesting. I took the project. What I remembered from biology, from what I was taught, that muscle is made of a bundle of fibers, and these fibers are made of a bundle of fibrils, and these fibrils are made of small proteins, and these proteins are like rods, and they slide onto each other, and when they slide, the muscle contracts. What I wanted to study is the contractility muscle, contractility of the muscle. However, most of the literature that I came across were black and white images of electron microscopy of dead tissue, not moving at all, and most of them were shown in terms of cartoons. It did not give me the kind of video or visualization that I needed for kind of muscle contraction. So for that reason, these are the black and white images that I was telling you, and those are those proteins that I was told that they were sliding on top of each other. So we took on a ambitious project to build a microscope to capture the muscle contractility live when the muscle is contracting in 3D. What that means is three-dimensional imaging as a function of time, and be able to capture fast dynamics. We employed cutting-edge technology, military-grade data acquisition cards, mirrors that were scanning in thousands of times per second, and mirrors that also literally shaped its form by applying voltage. All in all together in order to be able to capture the kind of fast dynamics that was going on when muscle is contracting, such as heart muscle. And when we turned that onto samples that were muscle, we saw a whole new world. Muscle was contracted, yes indeed. However, there were lots of dynamics that all of a sudden become visual. And those sliding theory was not satisfactory. It wasn't enough to tell us more. These sarcomeres, units of contractility, were not just sliding, but they were bumping into each other. There were all kinds of dynamics, chaos, everything was going on. It was life that was being visualized. However, with the microscope, we were able to see them in 3D, capture those dynamics while the contraction was happening, and look at them into the detail. So when we look at them into the detail, in the small regions, when we saw the contraction, in the small, sure, on the small scale, there were dynamics that were chaotic, unorganized, random. However, when they all synchronized, they showed predictable patterns and contractions. So using the physical model, we use that in order to explain muscle contraction using synchronization, using randomness, and it was no longer the sliding theory. So the old model of muscle, to me, that was given, were in black and white. In a simple case, when muscle is relaxed, it's just relaxed, not doing anything. These proteins are not doing anything. When it's contracting, it just reflexes, and it just takes another form. It's just binary. Either it's contracting or not. However, in the new paradigm, the way to explain it, well, let me give you an analogy, because I think analogies is our understanding. Imagine a room of children, full children. They will never sit still and not do anything at all and wait all of a sudden to switch in another form or another shape. They bump into each other, there's chaos. Believe me, I have a 2-year-old niece. It's quite noisy around there. And especially when there are a lot of them, all kind of things. But if you provide the right incentives, sometimes they do come together and show a predictable, synchronized activity that could be fun. And I hope that makes it understandable for all of us. With all, let me thank my group and everybody else that made this possible. Thank you very much.
Hello, good afternoon, everyone. My name is Autism. I'm a PhD candidate at the Faculty of Forestry. The title of my presentation is Hypothetical Advancement in Pulp Molded Products. Actually, pulp molded products are made from recycled paper. Any kind of papers, newspaper, magazines, fiberboards. The process for making this kind of products is quite simple. Just we need to dissolve recycled paper in a mixer, or we call it pulper. And then we send it to mold, and after dewatering, just we dry it. So it's quite simple and easy. And mostly we use this kind of products in packaging. The most common one, as you can see it anywhere in the market, is cartoons or cup carriers, or maybe you can find other similar products in the market. But the market for this kind of products is very limited. Actually, these kind of products are really environmentally friendly because they are recyclable, sustainable products, and compostable, and biodegradable, and also economically feasible because we don't need too much energy to prepare this kind of products. But these kind of products are very limited in the market. Why? Because of two main issues. The first one is the low strength. These kind of products, they can't carry high or heavy items. Just this for small items should be fine. Another one is the hydrophilicity of this kind of products. What does it mean? It means they absorb water greatly because we are making these products from cellulosic fibers, and cellulose absorb water. And when they absorb water, they become very weak. So there are some petroleum-based chemicals in the market we can add to these kind of products, and we make it water-resistant product. But they are not bio-based, they are not biodegradable, and not compostable. So we have innovated and now manufacture a bio-based binder. So this binder, we call it bio-binder. 100% we obtained it from a plant, 100% bio-based. And when we combine this bio-binder with the pulp or recycled paper, we can make a green hydrophobic pulp molded product which exhibits high water resistance and strength. And we can produce and sell this kind of product with a competitive cost. And the good things with this chemical, we don't need to change anything in the existing conventional pulp molding manufacturer. Just we need to add biobinder to the pulp. At the end, you have hydrophobic pulp molded products. And the final product is biodegradable and sustainable as well. So we are going to focus on two main or big markets. One is the horticultural industry, another one is the food packaging. In the horticultural industry, we are going to replace plastic pots in greenhouse industry with our biopots. Biopot is made of recycled paper and our biobinder. We can make this kind of biopot with different sizes or different shapes. And this year we sold about 300,000 biopots to greenhouse. And we can make with different formulation and different formulation with different degradation rate. And also we are going to enter food packaging industry and replace styrofoam plastic container with our green food packaging. So we hope with replacing any kind of plastic or styrofoam or synthetic polymers in packaging industry with our technology or our biobinder to reduce the greenhouse gases, to reduce our footprint, our carbon footprint on the earth, and also we hope to reduce the size of landfills in near future because our product is compostable, biodegradable, and sustainable. Thank you.
Hi, sorry. Okay, so I'm here to talk to you today about co-clinical trials. Co-clinical trials of autism in mice and humans, specifically changing how we treat autism. Autism spectrum disorder is a neurodevelopmental disorder characterized by impaired social interaction and communication, as well as restricted repetitive behaviors. Unfortunately, this video does not seem to be playing, but the little girl in the video is doing this. This is considered a repetitive behavior, which is one of the characteristics of autism. The prevalence of autism is really high. It's very common, and it's also a very heritable disorder. But it's also, unfortunately, very heterogeneous, which means that treatment for it is very hard. Current approaches tend to be very shotgun and just like treating symptoms rather than the actual disorder. And there's a strong need for, but lack of autism treatment would be individualized, where every single person would have a certain unique regime that helped them the best, whether that's drug, cognitive behavioral therapy, or dietary change, or something else. But we can't do this yet. So what we want to do is, as far as we know, the first attempt to bring co-clinical trials to autism to establish a translational paradigm for predicting responders from non-responders based solely on phenotype. We're going to do this by testing mouse models of autism alongside human autistic patients with several drugs and measuring various phenotypes. And personally, I'm going to focus more on the actual brain components and actual mice rather than the humans. This is a general outline for the basic pipeline of the study. Mice are going to be alive for a total of 9 weeks, with testing beginning at 5 weeks with the first of 3 MRI scans. Treatment ensues over the course of a month with the same drug that is being used in the human study, and on the last week of this month, the mice will go through a series of behavioral assessments. The study currently involves 3 strains of mice with their controls, all males with 28 strains, 28 mice per strain, half of which will be treated and half will be placebo. Now, what are we actually going to be measuring? The behavior. As I said, this is going to be during the last week of the treatment, and we'll be looking at 5 different paradigms used to test behaviors characteristic of autism, such as the repetitive behaviors except in a mouse model, and sociability. Behavioral tests is one way of measuring changes, but we're also interested in the actual brain volume changes. We can do this using magnetic resonance imaging. This gives us the ability to measure volumes in different regions of the brain to see what's different between the mouse models with and without treatment, as well as over time. So I've introduced you to the 2 metrics we'll be measuring in the project, but how are we actually going to be making changes? That is the actual treatment. Currently, we have 3 candidates for the study, and we are hoping to ultimately use all 3 of these in both mice and humans. First of which is oxytocin, a neuromodulator that is associated with social behaviors, giving us hope that it will help with the social deficits characteristic of autism. NNZ-2566 is a prodrug for insulin growth factor, which is a molecule that targets genes and proteins implicated in autism and leads to the growth of new neurons in the brain. Glycogen synthase kinase 3 beta is a fancy way of saying that the drug will improve energy metabolism and neuron growth. All 3 of these treatments target varying aspects of autism symptoms and etiology. So we are hoping to combine all of this data, both in mice and humans, to be able to take our population of ASD patients and stratify by responders and non-responders. Responders and non-responders for each of the different drugs and how they overlap, as well as based on phenotype, so we can ultimately take one individual and decide with his unique behavior, his structural phenotype, and genetics, what course of action is going to be best for him, creating truly individualized treatment for autism. And with that, I'd like to thank my PI committee, collaborators and helpers, and mostly Jacob Elliott and Taylor Bridge, as well as everyone at MICE. And also all of you and the judges, thank you for listening and for this opportunity.
Good afternoon, everyone. Today I'm presenting our cooperating research between Canada and China. So I'm from the Department of Applied Secretary and Human Development. So let's first imagine one day Einstein asked you, why does time go by faster when she smiles at me? What would you say to me? So you could be wiser than Einstein. You may say, forget about relativism. Just listen to your heart. You're a little bit faster when she smiles at you. So actually, this kind of wisdom advice is common in our lives. When we choose school, career, or life mate, we may seek for advice. Because we assume that there are some common themes in life. After all, Albert is only human. He has no different heart than us. So according to Berlin wisdom model, wisdom is defined as expertise in life, not in physics, not in psychology, but in life. So basically, they ask participants to give advice to some hypothetical figures on some life dilemmas, like talking to a teacher who feels as though life is meaningless because he has not achieved a dream he has since his youth. So they answer it, they will respond to this question, and then their response will be rated according by their criteria. There are 5 criteria: factual knowledge, procedural knowledge about life, how much you know about life, and how you conduct it, and a lifespan contextual, which means in different life stages, there are different themes, and also relatives of value and life priority, which means if you come from different society with different culture, you may value different themes. And finally, about the uncertainty of life. Like life is unpredictable. You will never know enough information for your life decision and how you handle it. So this is their criteria. So there is a critical of wisdom model because it ignores the emotional component of wisdom. But emotions are some experience vital for our life, and emotion is important for creativity. Without emotion, we will just like a machine and computers, not be creative at all. So wisdom shall be emotional. So now I have this study. So basically, they have 2 wisdom tests. First is on realizable dream. So I ask the participant to imagine one of your friends dreaming of becoming a singer, but he's really bad at it. If he asks you about his singing, what would you say to him? And the second one is the one I just mentioned. So a teacher feel like it's meaningless. These students, they imagine a camera as the eyes of the teacher and a friend and talk to him. And then their wisdom is rated by 5 trained raters according to the criteria. And also their facial expression was analyzed by our advanced software. It's based on Eichmann, Lie to Me, the American comedy. The character Eichmann is actually a real person. So this software analyzes the facial expression frame by frame. So for example, this is a frame analyzed by the software. Remember the camera captures 29 frames per second. So this is really, so many kinds of information in one video. So then finally, we need to average all the results of so many different kinds of emotion. It's not the mean value of the probability. It's based on this equation. I solve this equation and then give the average value of emotion because different person have different lengths of response. So we need to be, this result need to be averaged. So then we analyze the correlation between emotion and wisdom. So we found that the surprise is highly correlated with wisdom rating, especially in the first dilemma. So what is the meaning of a significant correlation? So for example here, you will see that the more surprised one feels, the higher his wisdom rating will be. So surprise is caused by the participant's own idea and thoughts because nobody talked to them. So this they do not expect. So this may actually contribute to their wisdom advising, especially in their more experienced dilemmas. So it reminds me that openness is believed to be more important than intelligence. There was no study to demonstrate it. So the take-home message is, it may be wiser not to be self-assured when advising about life, but prepare to be surprised by yourself. Thanks.
Hello everyone, my name is Peng. I'm from chemistry department. Today my topic is metal binding polymers for antibody-based cancer treatment. The therapeutic method based on antibody is one of the several most promising strategies for cancer treatment. This is because the antibody selectively target cancer cells and therefore minimizing their influence on healthy cells and healthy tissues. And one particular approach to further increase their therapeutic efficiency is to attach radioactive microions onto the antibodies. Because these microions can generate the radiation energies, and these radiation energies will lead to the dysfunction of cell nucleus and ultimately induce the death of cancer cells. But the antibody itself is not able to bind the microions or radioactive microions. So one relatively common strategy is to first modify the antibodies with some microbinders and use these microbinders to tightly bind the radioactive microions. These microbinders are usually some synthetic small molecules that can bind 1 to 2 microions on each of them. But one big limitation of this strategy is that the labeling efficiency of radiometals on antibodies is extremely low. Because most commercial available radiochemicals are suffering from low purity. They are accompanied with a large amount of non-radioactive microions, and they compete with those radioactive ones to occupy the microbinders. So the result is most of the antibodies, they were targeting the cells without bearing radioactive energy, so they cannot deliver these radioactive toxicities into the cancer cells. Therefore, in our research group, we are trying to circumvent these shortcomings by designing new polymeric microbinders. These polymers, they contain multiple repeat units, and each repeat unit was modified with one microbinder, and then we attach these polymers onto the antibody. In this way, we can amplify the number of microbinders on each antibody. So even though the purity of the radiochemical is low, we still can make sure more antibodies were actually labeled or carry these radioisotopes. In order to check whether these polymers are suitable for cancer treatment, we carry out a series of biological experiments to evaluate them. For example, we use some mouse model to see their distribution in the normal tissue and in tumor tissue. We also did some cell assays to see whether these polymers could carry these radioisotopes into their target sites. And we are currently at a stage to choose the best polymer and transfer them into clinical trials. In a simple word, we are trying to find the best polymer and to test them in the human body in the future. So I think this will bring me to the end of my talk, where I want to show my appreciation to my supervisor as well as all the group members in our research lab.
Hello everyone, my name is I'm a fourth-year PhD student at the Systems Control Group at the Department of Electrical and Computer Engineering at the University of Toronto. Today I'm going to present my presentation. It's called Maneuvering Control of Planar Snake Robots Using Virtual Holonomic Constraints. It's a joint work with Norwegian University of Science and Technology and TNU. So snake robots are a class of vehicle manipulator robots which are inspired by the structural characteristics of biological snakes. They can effectively be used for operations in challenging environments, and they pose significant motion control challenges because these robots use complex gates and motion patterns. One of the basic gait patterns through which biological snakes achieve forward motion is called lateral undulation. So here's a video of the robot performing lateral undulation. So during lateral undulation, the snake body undergoes periodic shape changes, and it looks like as if the wave travels backward on the body from the head to the tail. So in this problem, we investigate the motion control problem of maneuvering. We make the snake robot converge with some desired path and traverse the path with some desired velocity. We use visual holonomic constraints to solve this problem. So what is visual holonomic constraints? Visual holonomic constraints are relations between the joint angles of a robot that can be enforced via feedback control. They represent a paradigm shift in solving challenging motion control problems, and they can be effectively employed in robotic locomotion such as biped locomotion. Here's the structure of the control, the control structure that we use to solve this problem. Our controller has 3 stages of hierarchy. At the lowest level of hierarchy, we control the shape of the robot by using feedback control. We enforce the lateral undulatory gate on the shape of the robot. In the second stage of the hierarchy, we control the velocity vector of the robot by controlling the frequency of body oscillations and also the orientation of the head of the robot. Finally, in the last stage of the hierarchy, which is the path following controller, we determine a reference vector such that our robot follows that reference vector, and by following the reference vector, the robot converges to the path and traverses the path with the desired velocity. So here are the simulation results for a snake robot following a circular path. So as you can see, the lateral undulatory gate is enforced on the body of the robot, and the robot goes under periodic body shape changes, and it converges to the path and follows the path with the desired velocity. Thank you very much for your attention.
Hi everyone. Thank you so much for the opportunity to present my research to you today. So I'm Louise. I'm a chemistry PhD student from the University of Scarborough, and I'm going to talk to you about my research today. So in the medical world, there are a variety of methods that are used to look inside the human body. Some methods are more invasive than others. For example, you have exploratory surgery that is very invasive but can be very useful, particularly in cases where diagnostic is very difficult. Other methods, for example, computed tomography, which is also known as CT, uses ionizing radiation or X-ray, which can be very damaging to the human body. Most diagnoses today are done using magnetic resonance methods. There are a few different methods. The most common one is magnetic resonance imaging. You have an image of MRI up there. So magnetic resonance imaging being MRI, which looks at water protons inside the human body to produce images. A different method is nuclear magnetic resonance spectroscopy, or NMR. And NMR works in that it doesn't look at the water protons, it looks at specific chemicals inside the body. Both of those methods are really great because they're completely non-invasive. They work based on magnetic fields as well as radio waves, which is why people with pacemakers, for example, cannot undergo an MRI because the magnetic field will interfere with the pacemaker, and no one wants that. They're also really great because they provide 3D images of the human body. MRI can provide images of both the anatomy as well as the physiology of the human body. The first presenter, Sue, mentioned that she was using MRI for her studies in autism, so this is a perfect example of that. For example, another example would be looking at tumors. So MRI is great at looking at tumors. Like large tumors can be detected by MRI really easily. However, when it comes to cancer, early detection is really important. So if you want to look at early detection, you need to focus on biochemical as well as metabolic changes in cancer. So one of the most common metabolic change in cancer cells is that those cells are more acidic than normal cells. And in chemical terms, because I'm doing my PhD in chemistry, acidity is measured using the pH scale. In other words, the lower the pH, the more or the increased acidity. So the purpose of my research is to synthesize chemicals that can be used to detect pH by nuclear magnetic resonance spectroscopy, by NMR, so that we can one day detect cancer earlier and therefore save many lives. How do I do this? I actually designed a method called the slow proton exchange method, shown up there. How does that work? Well, the method is actually a lot more accurate than the common pH methods. One, because the signal is not dependent on the concentration of the probe, which is the case for most MRI-based pH methods. In addition, the method is ratiometric, which means that pH can be determined from the ratio of two different signals. This is actually very good, very advantageous, because it reduces errors coming from pH-independent factors, for example, the probe interacting with metal ions or inhomogeneities in the magnetic field. Now, SP1, which is up there on the slide, was the probe that I synthesized, and I used it to observe pH in bacteria. So by looking at the different signals of the protonated and unprotonated form, which is shown on the graph right there, I was able to obtain a specific pH. I tested this method in bacteria, E. coli, where I was able to monitor pH over time in the bacteria in the absence of oxygen, thereby showing that my method works. So hopefully today I shared both my method as well as the new compound that I synthesized. You understand a little bit of what I do. And in the future, we're hoping to move on to toxicity studies as well as animal studies, so we can one day bring that compound to the clinic and be able to detect pH as well as cancer earlier and therefore save a lot of lives.
Good afternoon, ladies and gentlemen. Welcome to my talk. My talk title is Stick the Tissue Together by Engineering an Elastic Surgical Glue. So my name is Yinan Zhang. I came from the Institute of Biomaterials and Biomedical Engineering. So this is my first year. So basically, they have two ways to stick the tissues together. One way is to using the suture. Another way is to use the crazy glue. It's called bio glue. So the advantage of using the suture is that it's very fast and very cheap and can be do it manually. So the advantage of using the glue is that it can stick the tissue easily. So, but the limits of the suture is that, like the Lee said, using the long tissues. The long tissues is very fragile and very floppy. It's difficult to suture those tissues together. So by using the glue, it can use to seal those holes, try to avoid the leakage. So this is the design properties of using the glue. The most important thing is to stop the leaking of the air or the leaking of the blood. So by the fast formation of the hydrogels or polymers. So these gels should be very elastic because this one will stick with the tissue and to form the contraction. Another thing is highly efficient in the blood environment. Because this gel should be used in vivo, it means inside of the blood. So it should be resistant from the blood. Another point is that this gel should be developed very biocompatible and biodegradable. And during the tissue healing, this gel is expecting to be absorbed by the human body. The last one is how to commercialize, to make it easy to handle. Yeah? So we want to inject this gel into the tissue and to make it form as a solid piece. So we don't want to stick this gel on the glass or on the gloves. So the aim of this project is to engineer an elastic biocompatible and biodegradable surgical glue with proper mechanical property from recombinant human tropoelastin. Why we use recombinant human tropoelastin? Because this tropoelastin is directly taken from a human protein and also it has good elastic properties. So this one is briefly giving you an idea how about this the physical and the mechanical property about this gel. So at the beginning we have the tropoelastin molecules, and then we methylacrylate it by the methylacrylate acid to modify the structure to form the material. It's called the methylacrylate tropoelastin. Then we shine the UV lights on this gel and to make it become solid. Then this gel has a certain elastic property. It can be extended more than 4 times of the original length. So before to do the in vivo test, we want to test this gels, how the adhesion gel behavior on the tissue. So we developed the wound closure test. So basically we have 2 pieces of glass. We have the pig skin and cut it like an incision, and then pour our gel on it, shine the UV lights, and to form the glue. So, and then we do the mechanical test on this glue. So the adhesive strength is present how strong about the adhesion force between the gel and the tissue. So by using the different concentration of the material, so we can achieve very high adhesive strength, which one is much higher than the commercial used Avicel and Tosin. To further validate these gels, we also studied the in vivo test by using the red lung model. So basically, we make the incision on the lung, and then we pour this gel on the lung to seal it and to test the burst pressure about this lung. We compare the healthy lung and with the incision lung. So basically, it shows almost the same result. As you can see, there's no significant difference, but when I compare the commercial use Avicels, it shows much lower burst pressure values. We also further intend this project to the big animal test. So we use the pig lung to repeat the same test. So surprisingly, as you can see, this one, the suture, is giving the very low value. Why? Because this lung is very floppy. By suturing this lungs, we make further invasion to cause the holes leak of the air. So by using the metro gels, which one is comparable with the FDA Pro gel, shows very good properties. So we can also use these gels to use for the small arteries. For example, like in this severe environment, it's very bloody, but by using these gels, we can get very good property compared with the healthy artery. So in conclusion, this project is interdisciplinary research by combining the material science, mechanical engineering, and the biomedical engineering. So an elastic, biocompatible, and biodegradable glue through the photolinking was successfully engineered. And this surgical glue can be easily commercialized, and its degradation and physical properties are controllable. At the end, I would like to thank my previous supervisor and my current supervisor. Thank you.